Welcome to another video, guys. Thanks for coming on. I hope my voice sounds good today. And this is going to be for protein synthesis. This is going to talk about how DNA, the letters, those bases, how that gets turned into a message for your body to make proteins that will make your hair color, your eye color, even things like your behavior, some people think. We're going to get going on that. I'm going to have you go into your packets for this, but I am going to stop a little bit earlier than when yours does. So we'll get to the rest of that in a different time. I'm going to try to make this short for you. So let's talk real quickly. Some of this will be review from things we've done in class, but most of this will have some pictures you've never seen, and so it's a good way to lecture through. And again, this is where you can come to me with questions so that there's things I don't have to do in class. But if you find something that's confusing to you, write that down, and then you can say, I understood this, but I didn't understand when you said this. So let's start quickly. Protein synthesis from genotype to phenotype. We're going to look at how we take the letters of DNA and turn it into what you're going to actually be like, what your proteins are. And you know this is called the central dogma theory. It basically just says that DNA can be replicated, but it can be transcribed into RNA, which gets turned into amino acids, and amino acids get put together to make proteins. What are proteins? So proteins are molecules. Macro just means big molecules that serve lots of different functions in the body. They can be pumps, they can be tunnels, they can be enzymes which help speed up or slow down reactions in the body. It can be your color of eyes, that's a protein that's made, color of your hair, etc. The building blocks of proteins are called amino acids, and they're coded for by your DNA. And of course, your ability to function depends on this DNA being coded correctly. And in case these terms come up in your book, amino acids, technically, the way that a scientist would think about it is that amino acids get put together to make these short little chains called peptides. And the peptides get put together to become what we call many peptides or polypeptides. And those actually get put together to make proteins. So proteins are actually made up of these smaller building blocks, but the ultimate source of proteins are the amino acids. RNA, as you know, is called ribonucleic acid. It's very like DNA. It has a sugar, a phosphate, and a base. The sugar and phosphate make the backbone, and the base sticks out like it does in DNA. So there it is. In case you don't know, this is the phosphate, PO4. This is the sugar right here. And this is the sugars with this ring structure here. And this is the base right here. Sugar, or phosphate here, sugar here, base here. Phosphate, sugar, base. And each one of those is still called a nucleotide. It's just called an RNA nucleotide. And RNA is different than DNA, as you know. The sugar is called ribose instead of deoxyribose. That's why the name is different. The bases are AGC, but no T in this. This is going to be a U, uracil, and it's single-stranded. So as we learned about in class, when RNA matches up with DNA, everything is normal except that wherever a T would be put in for RNA, RNA doesn't have T's, it only has U, so you put a U instead. And here's a picture of kind of what that looks like. Here's that ladder, that twisted ladder. And this is kind of RNA. It's like a ladder that has the side rails missing, so it's only single-stranded. It doesn't have that other side with the backbone in it. And there are different kinds of RNA. The kind that we really talk about a lot is called messenger RNA. It's the RNA that carries the DNA's code out of the nucleus. Remember, the DNA's stuck. There's a second kind of RNA called tRNA, or transfer RNA, and that actually is the one that has the amino acid on it, and it kind of brings the amino acids over at the ribosome and helps to form the protein. I'll show you that in a picture. You've learned this, but when you take the DNA and you turn it into an RNA sequence of messenger RNA, that's called transcription. And when that RNA sequence goes out to the ribosome and the amino acids get linked together to make proteins, that's called translation. And we did some kind of secret code activities to do that too. So what's transcription? That is when you take DNA and you make messenger RNA, and DNA unwinds, as you've done before. You've seen it like unzip, and one strand serves as what we call a template, and then the RNA nucleotides come line up. G with C, but wherever there's an A, RNA will come with a U because there's no T in RNA. And that's called the complementary strand. So for example, if the DNA went ATT, GCG, etc., this would be A, the U would come here, the A would come and match up, and then this thing would be glued together into this long single chain sequence called messenger RNA. So here's a picture of that happening. This is in the nucleus, and you've got your DNA is this blue double spiral thing, and here's the RNA forming. The orange backbone is different because it's ribose instead of deoxyribose, and then you can see there that there's U's instead of T's. So there's a U there, there's another one coming. This is all happening in the nucleus. So remember that RNA acts as that messenger for DNA, and it gets it out of the cell and communicates those instructions back at the ribosome. So at the ribosome, we have translation, and this happens where the messenger RNA sequence that was in the nucleus comes out and lays itself down on the ribosome, and the tRNA comes along and puts together the amino acids that are needed. So it tends to be called the translator. There's two kind of sticky parts to it. One part has the amino acid on it, and one has 3 letters that match up with the messenger RNA letters. So this picture shows this is the long messenger RNA at the ribosome. This white big thing here is the ribosome. This is the messenger RNA that came from the nucleus. And here's a tRNA. Notice it has 3 letters that will match up with the messenger RNA letters, and then attached to that it has an amino acid. Here's another one right there. And then as each one comes, the amino acids are linked together into this like beaded chain looking thing that's called a polypeptide or a protein. So a codon is technically considered, as we talked about in class, to be the group of 3 messenger RNA codes. And then the anticodon is considered to be the one that's attached to the tRNA. I can go back a second as I'm thinking about it. Let's go back to one more slide just to kind of show you. So this is the tRNA, this is the sticky side, this is called the anticodon, and this is the codon of the messenger RNA. Codon, anticodon, transfer RNA, and then on the other side is that sticky part that's got the binding site for the amino acid on it. And then we'll practice with this code, but you use this code chart, which I'll give to you on a test, to figure out what amino acids it is. The amino acids are abbreviated there with PHE and LEU. You don't have to know the names. And basically you go to look up something like, for messenger RNA, let's say you were looking up CUU. You'd go to C here, that's the first letter. You'd go to U, which is the second letter, and then you'd go, for the third letter, if it was CUU, you'd go to U as the third letter here, CUU, and that's called leucine. We're going to do this in class. So when you read the code, you find the sequence of 3, you put the first nucleotide on the left, the second one goes on the top, the third one goes to the right, and then you identify the amino acids where the 3 meet. And again, I mentioned this in class, but there's always one codon that says start the protein, and then there's a few that say stop. That means stop making that protein because we're going to make another one now. And there are many, thousands in some cases, of different proteins made from each chromosome. Translation is very important because that makes sure the protein's correct. If it doesn't have the right amino acids in the right order, something's going to be wrong, and the protein is going to be deformed or won't be formed at all, and it won't work and do its job. And this is a final picture that I want to show you. This is the whole cell membrane there. Here's the nucleus. This is where the DNA unzips. That blue is the backbone of the messenger RNA that's forming there. That's called transcription. That leaves the nucleus, comes out to the ribosome, and then here's that tRNA. This is the anticodon, and these are the amino acids linking together to make the protein. So that's called translation. So the 2 steps, transcription, translation. Okay, I think that is enough. Whoops, we're not going to do mutations. We're going to talk about those in class, but we're not going to do mutations right now. So when you get to this part of the slides, you can go ahead and stop it, and we'll talk about mutations at a different day. See you later, guys.